Hello, tonight we're going to talk about the chromosomal basis of inheritance. This is really fun stuff. Let's go. According to the chromosomal theory of inheritance, genes have a specific location on chromosomes, and that is the chromosomes that undergo segregation and independent assortment. So, what we know is that there are chromosomes, and genes are on chromosomes, and chromosomes segregate and independently assort. So what does that mean? Well, what it means is that when two people come together and make an offspring, when that offspring then makes their own DNA, you get a mixed up combination of the chromosomes. So you can have some chromosomes from one parent, you could have some egg or sperm that have chromosomes from another parent. Further, on those chromosomes, the genes independently independently assort from each other. Now, we learned all of this by using a special organism called a fruit fly. Fruit flies are the organism of choice, first of all, because they only have 4 pairs of chromosomes. So there's not as many chromosomes that you have to look at. And they can mate and produce fertile babies who then can mate every 2 weeks. So every 2 weeks you can have a whole new generation of fruit flies. That's really cool because in a very short period of time you can do some studies and find out all sorts of information. Also, they produce hundreds of offspring at a time, not just 10 or 20, hundreds. Some of the characteristics that people have studied would be looking at the eyes. They're called barred eyes because they're an odd shape. This is the normal fruit fly. They could have wings that have a strange cut shape to them. They could have rudimentary wings, an abdomen that is rotated, vestigial wings, which are they can't fly with, curly wings, bithorax wings, so one set is weird, the other set's okay. You could have dichate wings. Look at those, very strange. So those are just a few things. They can look at eyes. They can have white eyes. They can have brown eyes. They can have all weird things. So the fruit flies are kind of cool to work with. From experiments on fruit flies, they discovered that some traits are found on sex chromosomes. In humans, these things are red-green color blindness, hemophilia, things like that. There are different genes on the, there are lots of genes on the X chromosome. There are very, very few genes on the male Y chromosome. Now, the reason we look at sex chromosomes is because in males, because they only have one X chromosome, they have an X and a Y, and they only have one X. Any genes that are found on the X chromosome, any mutant genes, they will have that characteristic. They will have that disease or characteristic or whatever it is. In girls, because they have 2 X chromosomes, it has to be found on both X's for the girl to have it. So it's less common for girls to get sex-linked diseases. We also found something called linked genes by looking at fruit flies. Linked genes are genes that are very, very close together on a chromosome, and they tend to be inherited together. For example, red hair and freckles are linked. They tend to be inherited together. Now, it's not 100% of the time. There are people who have red hair and don't have freckles, but it's much less common because they're right next to us on the chromosome. And so when crossing over occurs, it usually doesn't occur between them. They're too close together to have crossing over occur between. That's what a linked gene is. Okay. Here's a test cross between 2 different fruit flies. A wild fruit fly, which is the typical one, and then a double mutant fruit fly that has a black body and has vestigial wings. Okay? When they crossed them, the F1 wild type has, these 2 were crossed and created this one. And then they crossed this one with the same type of offspring as the first parent. And these are the different genes that they get. They get a 1-1-1-1-1 ratio. Is what they expect. But it didn't happen. Instead, they get a lot more of these two than of these two. Why? Well, the reason is those genes are linked. So this is an example of how they could use fruit flies to figure out linked genes. We then use linked genes to help figure out where chromosomes are, or where genes are on the chromosomes. Now, they created an arbitrary unit. It's called 1 map unit, which is equal to 1% of the recombination frequency. So if something crosses over and recombines, that's what recombination frequency means. If that were to happen in 20% of the offspring, then it would be 20 map units. Okay? That's how you figure it out. Now, a map unit really is the distance from the centromere on the chromosome to the tip of the chromosome. It doesn't give exact distances. It only gives kind of like the order of genes on a chromosome. So this is kind of a good example. This is the type of question I have seen on AP exams before. So this is kind of a logic problem. Well, let's take a look at it. Okay, you have 4 genes, J, K, L, and M, on the same chromosome. Given the map units between K and J is 3, K and L is 8, J and M is 12, and L and M is 7. What's the order of the genes on the chromosome? Well, you need to figure that out. I'll give you a couple of minutes. Pause the video for a second and figure it out. Now, I gave you a hint. J must be near K because they have a very small map unit, only 3. And J is near M, or excuse me, J and M are the farthest apart because 12 is the biggest number on there. So figure out the order of these. Now, the next slide has the answer, but let's give you a minute. Did you cheat and not do it? Come on. You're supposed to do these problems. Okay, here are your choices. Was it JKLM, JKML, KJLM, KJML, KLJM? Which one of those did you pick? Now, it can be reversed. Could have all started with M or with L. Correct answer is C. What you do is you kind of draw a number line. You place the two closest together, very next to each other, and then you start adding in the other points. Keeping in mind, they may very easily give all the answers starting in the reverse order. I kind of like that problem. Okay, inactivation X is not a disease. Inactivation X is something that happens in all females. Now, in a female, when you were 8 to 16 cells big, so you're not very big. The cells have divided a couple of times. What happens is one of the 2 X chromosomes that you have condenses and becomes something called a bar body. It becomes inactivated. However, you're 8 cells big. It's not always the same X chromosome in each of those 8 cells that gets turned off. Sometimes it's mom's X chromosome, sometimes it's dad's X chromosome that gets turned off. It varies. Well, that's not usually an issue, but if one of the X chromosomes happens to have some characteristic and the other one doesn't, then some cells in the body will have that particular characteristic and other cells won't. So it kind of makes women a chimera, which means although we have the exact same DNA, it means some cells in the body have different DNA turned on where others don't. So it's kind of an interesting little thing that happens in girls. It's weird. I get that. It's kind of weird. It's not really understood really, really well, but you can see some of the results of it by looking at calico cats. Calico cats are always female, and this is an example of that. The different patterns that happen on the calico cat are caused from which X is activated. The yellow pattern would have been one X that was activated. The black pattern is a different X that is activated. And so you can see on this particular calico cat the differences of which cells derived from which cells in the body, in that cell that, or in that organism that was 8 to 16 cells big. So which one derived from which? And you can see how we're just a chimera, a mixture of the two X chromosomes. It's kind of interesting. Again, not terribly well understood, but it's an interesting phenomenon that happens in girls. Okay, some terms that you need to be aware of. Non-disjunction, I'm going to talk about several times. Basically, non-disjunction means that during meiosis, the chromosomes do not separate properly, and as a result, the offspring, or the sperm or egg to begin with, will have too many chromosomes. Now, that leads to some pretty nasty diseases. First of all, the term aneuploidy just means an abnormal number of chromosomes. So in a human, you're aneuploidy if you have 45 or 47 or 69 or 92. Okay? Trisomic means you have the normal 2n number plus 1. So a trisomic individual would have 47 chromosomes. Something like Down syndrome is trisomic. Monosomic means they have the 2n number, which is 46 in humans, minus 1. So they would have 45 chromosomes. An example of that would be a disease called Turner's syndrome. Polyploidy means you have a multiple number of 2n. So you would have 2n, 4n, 8n. In mammals, polyploidies usually don't survive. In plants, they do. And there are quite a few plants that are tetraploidy with a 3n number, triploidy with a 3 number, tetraploidy with a 4n number. For example, the big strawberries you get at the grocery store are tetraploidy. They have a 4n number. If you would look at wild strawberries, which are small and about the size of an eraser on your pencil, those have a 2n number, but they're all derived from the same organism, the same strawberry plants. Somewhere along the line, there was a genetic mutation that caused a strawberry to grow that had a tetraploidy number, which caused the strawberries to be much larger. And then farmers took advantage of that and planted those seeds, and that's how we have all the strawberries we have today. Okay, it wasn't in the laboratory where we genetically did this. This was just an accident of nature, and it happens in plants frequently. Okay, the next are some, I'm going to go through these really quick because I'm going to talk about them again in a later PowerPoint. Some genetic mutations that can occur to chromosomes. So these are full chromosomal mutations. Okay, first one is called a deletion. If these are the genes on your DNA and D is deleted, then this is what you would have afterwards. A duplication is when this is what you started with and you duplicate or copy these genes, so now you got multiple copies of them on your chromosome. An inversion would be when these genes are cut and re-put back in, but in the reverse order. Translocation is when some genes are cut from one chromosome and attached to a completely different chromosome. Now, you can have a reciprocal translocation where on this chromosome it's going to be attached to it, the end is cut off and reattached to that one. Or you can just have a translocation where it just happens on this one and this, and then it goes to this one. Translocations are kind of bad because what happens is those genes that are translocated to the wrong chromosome, in essence, are lost to the cell because it doesn't know where it put them. It can't find them then when it needs them. They're supposed to be on chromosome 5 and they're on number 17 and it doesn't know that. The cell doesn't keep track. It just looks for them where they're supposed to be and they're not there. So translocation is kind of a bad one. It's also called a jumping gene where a virus can cause this to happen, and it attaches and then takes genes with it when it jumps to a new chromosome, and so it's kind of bad. That's it for tonight. Have a wonderful night.